Hello everyone, and welcome to our Bio2Lab on Ancestry Inference. So today's lab is a puzzle solving lab. And in order to get the most out of the lab, I'd strongly recommend that you read ahead, see what you can solve on your own, and then use this lab video as a means of trying to fill in the blanks. I promise you that if you just watch me solve these puzzles, you won't remember them well enough or be able to solve them on your own come time for the exam. Therefore, the way to get the most out of this is read on your own, see if you can piece it together, then use this video as supplementary material. So with that disclaimer said, let's go ahead and take a look at what kind of puzzles we're trying to solve. The lab today is looking at haplotypes. We use haplotypes, just like we discussed in the lecture on biological anthropology, to try to piece together relatedness in populations. Not only within populations, but we can also use this between different species. You may have even heard statistics like we are some large percentage similar to chimpanzees or some small percentage to polar bears, or even further away to plants like corn. Ultimately, the way in which we study relatedness is with the alignments that we've used before, and then seeing this series of alleles that occur on a chromosome within a population. So gene alignments allow us to establish relatedness across species. Haplotypes do much the same within a species. We look at haplotypes in the context of our genotype data. As we established in our last lab with alignments, we'll recognize that most of the genome between two humans are fairly similar. But the places where there are differences can be hugely informative as to how relatedness is structured across a population. We therefore need a large treasure trove of single nucleotide polymorphisms, or SNPs, that are oftentimes biallelic, most of the time biallelic. In fact, in my own PhD work, we discarded any SNP that was not biallelic. We assumed that there was something weird going on with it, and that we were not going to use it for our analyses. Biallelic simply means that there are two different versions. And so we can now, with an understanding of how genes look in terms of their FASTA files and how to perform alignments with some of those sequences, we now can extrapolate this information and get rid of all of the noise around it, the stuff that's not informative, and really focus on only the informative pieces. So what you'll see in this table are two different alleles at 1, 2, 3, 4, 5, 6, 7 different sites. So this is not a case where we have 7 base pairs, where we have an A next to a C, next to a G, next to an A, next to an A, next to an A, next to a C. Note there, I was just taking the first letter listed here. These positions are thousands of base pairs apart. So what we're doing when we look at these haplotype maps is we are figuring out which SNPs are informative and only looking at the information there. Everything in between this A and C is either not informative to our haplotypes or exactly the same. They match 100%. So we don't need to look at those regions. So these SNP ID tables, and again, I copied this directly from maize data. So this is actually an output from a file looking at the haplotype map for maize, is just keeping the informative bits. Now, if you think that we're cutting out huge sections of the genome, you are right. There are large regions that don't have these markers or SNPs, but we still have 55 million single nucleotide polymorphisms, or SNPs, across the maize genome, just looking at these heterozygous sites. So yeah, sure, you can think that we're getting rid of a lot of data, the 3 billion base pairs in the maize genome, whittling it down to 55 million, but that's still a huge amount of information in order to establish relatedness. So my first task for you here is to establish what is the reference allele. When we're looking at these tables, the reference allele is always the first one. So in this space, you just need to fill in those first alleles, just as we did above. The A, the C, the G, the A, the A, the A, and the C. And this is what I mean by you definitely should be pausing the lab video and trying to solve things yourself before I explain them. It's fairly easy once you see it, but part of the gift of an education in biology is being able to puzzle solve with the clues that I give you in the text. And I do think that there are enough in there, but use the lab video as a crutch. So for visualization purposes, in order to look at this haplotype, we oftentimes paint them horizontally. And I do mean paint. Today, you will be painting haplotypes. Our reference haplotype is A, C, G, A, C. And we're going to go ahead and paint that in our green color. Our non-reference haplotype, in this case, is the Missouri 17 or Mo 17. In order to see what the markers are at the Mo 17 haplotype, all we got to do is look at the non-reference alleles. So go ahead and pause, go back to that table, and see if you can write out your own non-reference or Mo 17 haplotype. Now, I'll draw attention to the fact that these are not the complementary bases of what we see in the reference allele. For example, the first reference allele is an A. We know an A does not pair with the C, so know that the reference, non-reference are not complementary bases. They are mutations. They are polymorphisms. Somewhere along the line, whether A was there originally or whether C was there originally, we don't know. But one mutated into the other, and now we have this heterozygous site. So let's see if we can start to paint these separately. I'll go ahead and paint the non-reference, the Mo 17, in black. So we've got black and green as our two colors. Now, we're able to impute or deduce additional information from these sites, even if we're missing data, based on what haplotype they're found. Because you know that in order to originate from a B73 haplotype or a reference haplotype, you're going to inherit all of the markers in between, unless there's been recombination events. So we are going to be operating on the assumption that you're going to get likely one, at most two recombination events in a single haplotype, particularly for this lab, and therefore missing pieces can be imputed based on the context of the paint around them. So what exactly do I mean by this? Well, let's paint our individual here. If we are looking at this new individual, they have an A at their first marker. Was this a reference or a non-reference? If you don't have colors, you can go ahead and use numbers, but looking at the reference haplotype, we see that an A is our type 1. We see that next up, a C is our type 1. A G is our type 1. And then at our fourth marker, an A is our type 1. But then, at our fifth marker, we have the non-reference type. Therefore, we paint it with a 2. The next is also a 2, and the last is also a 2. This means that we have encountered recombination somewhere between marker 4 and 5. Everything behind the recombination event is assumed to be marker 1, and everything after the recombination event is assumed to be marker 2, based on haplotype imputation. So what do you think is going on with this individual? Well, I think I answered it for you. A recombination event has occurred between the B73 and the MO17 haplotype. Recombination is an essential process in breeding. So next up, we'll paint a lower coverage genome, meaning that we don't have as much certainty in our SNP calls. So we're not as certain about any individual marker, but we can hopefully use imputation to help us correct any mistakes that we might find in our original sequencing. So let's go ahead and paint this individual. Pause the video if you get a chance, otherwise I'll just go on forward. The A is a reference, the C is your reference type, the G is our reference type, followed by an AA, reference type, with not a 1 or a 2. The G in our 6th position has neither base, or neither haplotype. It is neither a 1 or a 2. But then we look at the C at the end and we see that it's a 1. So, with a low coverage genome, is it likely that this was a mutation or something else went wrong? What could you infer about what is the likely allele that we should have encountered at this 6th SNP? Well, if you concluded that this likely should have been an A because the rest of the haplotype is B73, well then you're starting to get it. The thing that's different about this haplotype is its lower coverage. We've got a non-canonical or non-traditional allele call in there, likely due to the fact that the low coverage or low confidence in our calls led us to make a bad call. So imputation can help autocorrect some of these issues. Think of it kind of like when you try to spell your last name and you throw in an extra letter there. The computer is able to recognize the fact that that's not how this individual usually spells their last name. I can probably autocorrect it so that way it matches the typical thing that they're writing in that space. Imputation kind of operates on a similar framework as spell checks. But this time, you're checking sequencing. It also means that we can sequence things to low coverage, have some bad calls, but then impute them to fix them. So we don't have to have thousands of dollars in order to sequence every single corn individual. With imputation and a knowledge of the parental haplotypes, we can start to deduce what markers they have at different locations. But what happens when we throw a monkey wrench in the system? And I will encourage you to try to figure out what's happening with this. I promise you there are all the clues that you need in this table to try to figure out these new additional haplotypes. Hopefully you paused the video. If so, welcome back. Let's try to piece this together with our 2 new markers, the HHA01 and HHA02. So we know what the B73 haplotype was. We know that, and I'll write this down here, so zoom out a little bit. We know that the B73 haplotype was our A, C, unknown, unknown, G, A, C. This is what we see for our B73. Now, we can figure out what was going on with those 2 unknown inserted marker types. Does B73 have the TC or does B73 have the GG? Let's read our paragraph. For example, so haplotypes also often have a shared history, meaning that haplotypes will share SNP variants. For example, let's say that B73 produced thousands of offsprings for many generations. All those offspring will share its haplotype, but also have the ability to mutate and evolve over time. One of those offspring happens to be well adapted to drought. Deep sequencing, so now we're sequencing to high coverage, of this new individual shows that it matches the haplotype of the reference, but has two novel variations at these two new SNPs. Mo17 does not differ from the reference at those two SNPs. So we can go ahead and see that our reference type, B73, is still our first two. So we're going to write out our TC here. And when we're looking at Mo17, because Mo17 doesn't look any different, we know that the TC will be shared between Mo17 and B73, but then that the rest of the haplotype is of the non-reference form. So we still have the C, the G, just so we're clear here, the C, the G, then we're TC, and then we're all non-reference, meaning that we are T, T, G, T, G. So how about our new individual, PH179? These are all real names for corn varieties. Pioneer Hybrid 179, B73 for Iowa 73, and Missouri 17. Just accession numbers for these. Why I still remember that? Not certain, but it stuck. PH179 is going to be of the B73 type. B73 was its parent here. Therefore, it's got AC. But then in the two new sites, it's got the non-reference haplotype, GG. Following along, we go back to reference. G, A, C. And so now, translate these down into there, you can start to piece together which new varieties might be drought tolerant, assuming that this space here determines drought tolerance. You now can have a situation where you're painting new, or rather all reference, where we're painting non-reference, but shares with reference, and then where it is all reference, except for in this new region. So you're able to piece together, if you have a TC, this region would mean that you are in a B73 haplotype. But if you've got a GG here and the rest of it's the same, you know that you have the PH179 type. And with that in mind, I want y'all to go through and try to paint all of these haplotypes. In some cases, you will have Ns. Those are unknown bases. See if you can or cannot impute in these different areas. I won't go through solving these on the video, but I do encourage you to contact some of your classmates and see if y'all got similar results, and if maybe y'all can argue about this to see whose explanation makes more or less sense. This is the kind of thing that you'll have to do in lab meetings or team meetings at a company. You have to get good at defending why you make these decisions, so please do so. For our last question here, it asks, how is it possible that when sequencing a single individual of corn, we can have both the TG alleles at HHA1? So looking back at it, how can you have both this and the CG allele at HHA02? Well, how do you have both SNPs? I'll write one word and hope you can solve the rest. So, enough with the corn, and now to the people. We can use haplotype painting to try to piece together human ancestry. However, when you look at the nuclear genome, the evolution rate of DNA within the nucleus is fairly slow. Human populations have expanded drastically over the very recent past, something within the last, say, tens of thousands of years. Therefore, nuclear genome haplotypes may not be informative enough to accurately piece together human migration patterns. Fortunately for us, there's another separate container of DNA that mutates faster and therefore gives us a better molecular clock over our more recent human history. This, of course, is the mitochondria. These organelles are maternally inherited, so we can actually trace your maternal line based on the haplotypes in your mitochondria. Additionally, mitochondria have circular haploid genomes. Haploid genomes, in contrast to some of the other genomes that we talked about above, are not heterozygous. This means that we can more clearly piece together connections. And so, trying to look at the history of all of human migration and the entire mitochondrial genome, we see that the mitochondrial genome is much, much smaller than our human nuclear genome. Now, this is in part because some essential mitochondrial genes have actually popped over to our nuclear genome, leaving the mitochondria with less than 17,000 bases in total, only 13 coding genes, 24 non-coding genes, and most of that related to processes of cellular respiration, like genes coding for coenzyme Q, or enzymes that work with NADH, as NADH is one of the essential components that mitochondria will break down for energy. So it's really mostly housekeeping stuff that's present in the mitochondria, very few genes, and therefore a lot of space for mutations that don't have an impact to accumulate. And so that's really, really important to our mitochondrial haplotypes. Most of the mutations that are occurring in the mitochondrial genome don't have a phenotypic effect. They don't mean that this mutant is functioning differently or bad or worse. They just tell a history of how things are related. This means that there's no disadvantage or advantage to having these mutations. It's just, again, a way to trace who was more closely related to who, in the same way that we were able to trace haplotypes with new variants in our corn example. So, here is our image of mitochondrial haplotypes. The graph operates from a top to a bottom, and we see that there is some mitochondrial Eve haplotype, an L type, that has diversity of types from L0 to L1 through L6. And you'll notice, look at how different these are. On the x-axis, or the distance of this line here, is the amount of diversity contained between those different haplotypes. So L0 is very, very different from L1 through 6. And then this tremendous amount of diversity is what we see between L1, L2, L3, L4, L5, and L6. These very distinct, very diverse haplotypes. Now, M and N are two different haplotypes that descend directly from L3. So you have this inheritance pattern where M and N will be very, very similar to L3, but have their own markers that distinguish themselves. In the same way, you get this cluster of M-type haplotypes and N-type haplotypes, with distinct groups in the OAS, IWXY, and then a large amount of diversity within the R type. So you can look at this graph or image, phylogenetic tree, as kind of a family tree for human history. Now, this doesn't mean much unless we start painting where on planet Earth these different haplotypes are found. Because this, in theory, should give you some idea about different migration patterns. If you see a K type coming in distinctly amongst a whole bunch of M's, we know that that K haplotype had to move there via migration, and where is the nearest source of those K haplotypes. So we can stare at this image for a bit. And again, just like before, I want to encourage you to try to solve this on your own, to see if you can trace these patterns and trace these diverse mitochondrial haplotypes to see if you can answer the questions in the lab. Those being, where do we see the origin of humans? Where are those really, really diverse L-type haplotypes? Where did migration go through? And where are these very distinct haplotypes splitting? When did Europeans and Asians split apart from one another? So hopefully you paused the video. If not, I'm here to kind of tell a story. You can additionally gather some information here from the age and years. You'll notice coordinated arrows here. You, of course, should notice an L-type haplotype, the mitochondrial Eve, that then separates differently between the L0s that traveled south and the L1, 2, 3, 4, 5, 6 that traveled north. So that first split in our graph, like all that diversity, is a north-south split. The L0s, ancestral populations that traveled south, L1 through 6, ancestral populations that traveled north. Crazy how that isolation by distance can cause so much distinction between populations that we can detect it even 200,000 years after it happened. Now, from our L3 haplotype, you'll notice that the next breakdown we're trying to follow is our M and N, and we see that this travels through kind of the Middle East. Some hypotheses suggest through Saudi Arabia, but we see all of our M, N, and R haplotypes here in the subcontinent of India. So this will include the haplotypes present in the Fertile Crescent. Though again, these migrations of humans are 100,000 years before agriculture happened. So don't think that we're yet anywhere near some of the ancient civilizations like the cities of Babylon or the Sumerians or the like, that these would have been migration patterns that occurred well before agriculture and quote-unquote civilizations occurred. Now you'll notice that M and N split here, but that the R type was a subtype of N, so we see that arrow happening here, somewhere between the last 90 and 55,000 years. And then after that, we see these migrations where, hopefully you notice, the split between the Europeans and a lot of our Asian populations occur. Something in the 55 to 30,000 years ago, where the M and the N types travel either to central China or Southeast Asia, colonize these land masses here, Southeast Asia, that would have been connected, so you could have walked from one to the other, or travel into China and disperse from there. Now, at this point, there would have been other humans living in these areas, not Homo sapiens, Homo erectus, Homo neanderthalensis, a new species being discussed more and more, the Denisovans, and even potentially some Floresiensis, a smaller in stature human species that we found remains of on the Southeast Pacific Islands. Now you'll notice that this group in China does disperse centrally within the last 30 to 10,000 years, that this line traces all the way to the Americas. So in this way, we see haplotype sharing between East Asians and Native Americans. Gives you an idea about the origins of those populations. Now, they would have spent a distinct amount of time, distinct maybe not the right word, but a significant amount of time on this mammoth steppe of Beringia here, something not captured in the graph, but something that we today are continuing to explore the implications of. So we have our answer to the question of which populations do Native Americans descend from in looking at our East Asian haplotypes. Now we can look at some of our European haplotypes and see that a lot of European diversity is descended from that R haplogroup that split apart something like 55 to 30,000 years ago. We do see some migration in of more disparately related haplotypes like the H haplotype, or rather some more developed haplotypes within Europe, these ancient H and V haplotypes, and some re-migrations, so some new migrations from the Caspian Sea or thereabouts. But that is certainly a unique circumstance in Europe where we get seemingly waves of migration that kind of block out previous haplotypes. But I mention H and V because my wife is actually of a V mitochondrial haplotype, an ancestral, really old school, older than the more recent migration event, haplotype that is oftentimes referred to as Old Europe versus the New Europe, which my mitochondrial haplotype is of the New Europe type. Those Proto-Indo-Europeans, likely. Now, we have another way to look at this graph in terms of relatedness and distance. So you have a phylogenetic tree whereby we see clustering of these different types of haplogroups. So the N's being a subset of the R's, the M's being quite diverse, and we see some of the additional letters associated there. But what you'll notice is where do you see the vast majority of the diversity? Is actually in those L types. The lines, the numbers associated with each of these, the calculated genetic distances, are the absolute greatest in Africa. And that is where we're going to be answering our first question. Where did humans originate? Well, we know from the map that it was Africa. What evidence do we have within the mitochondria to suggest that humans originated within Africa? Well, what causes mutations? What causes new haplotypes? Mutations are predictable over time. Therefore, the more time a haplotype has been around, the more mutations it would have experienced. Again, they're not positive, they're not negative, they're just there. They're telling a history. And therefore, the places where we see the most mitochondrial diversity, we assume humans have lived the longest, and therefore are where we originate from. Relatively speaking, some of these really old European haplotypes have a very small genetic distance between them when comparing to the massive genetic, and if I was to trace this line, L0 to L1, L6, the length of that line is significantly longer than the length of our H and V mitochondrial haplotype lines. Length of the line, of course, corresponding to genetic diversity. So that gives us a good idea of where humans originate and when we split. We talked about the Native Americans and their shared ancestry with the East Asians, as well as those native Australian Aboriginal populations and some of our Southeast Asians. And then looking at the last continent to be populated, the most recent, though not the last continent to be populated, of course, being the Americas, particularly the southernmost tip of South America. But you can see that only within the last 10 to 1,000 years, actually some of these very distant islands in the Pacific, like Easter Island or Rapa Nui, would have been colonized within the last 1,000 years. So it's kind of incredible. And then we see that these migrations to the Pacific Islands coincide with a colonization of Madagascar. Despite the fact that Madagascar was right next to Africa, humans did not arrive on the island up until the last 1,000 years. That, for explanations of that, you can look at ocean currents, but that is well beyond the scope of what we're talking about here today. But I will encourage you to look into it. It's super interesting. So you can additionally answer this question on greatest mitochondrial diversity. And in addition to greatest mitochondrial diversity, we also see the greatest nuclear diversity within this geographic region of human occupation. And then I want y'all to think about this one. Do our mitochondrial genomes show evidence of mating with Neanderthals? So I'll pause here intentionally. What do you think? Well, Neanderthals are adapted, or were adapted rather, to colder climates in Northern Europe. And since we can trace all human mitochondrial types back to Africa and no types back to Europe, to me that suggests that our lineage is unbroken back to this shared ancestry with a group that we call mitochondrial Eve. And I will, as in the lecture, emphasize that this is not a single person. This is a group of humans who would have shared the same mitochondrial haplotype. Not a single person, it's a group that share that haplotype. So, since we can trace our ancestry back to a single haplotype, not distinct haplotypes, and that ancestry is traced within the last 200,000 years, Neanderthals separating from Homo sapiens well before 200,000 years ago, we can assume that we have no mitochondrial evidence for mating with Neanderthals. But then, of course, looking at our nuclear genomes, whoa, we can have huge percentages, greater than the average person, somewhere between 1 and 3. We can go upwards of 5% being Neanderthal-derived DNA. What implications that has for human phenotypes and development? Still being discovered, but for whatever reason, mating did not occur or did not produce viable offspring that still survive to this day. It's possible that they did produce offspring. Maybe they weren't viable. Maybe they just, by random chance, did not continue on. But we currently believe that mating between a Neanderthal male and sapiens female produced offspring, but mating with a sapiens male and a Neanderthal female, well, we don't have evidence of that in our mitochondria at the very least. And we can't really tell with our nuclear genomes. So that's the best evidence that we have for now. Now, mitochondrial ancestry does not always agree with human ancestry based on the Y chromosome. Is that okay? Well, where do you get your Y chromosome? For that, hopefully you know enough about human genetics to know that the Y chromosome is male-derived. Do your parents, your mom and your dad, share the exact same history looking back? Well, probably not. They come from different places. They tell different migratory stories. So it's perfectly okay that our mitochondrial ancestry does not agree with our Y chromosome ancestry. They are, after all, inherited very differently. Now, in order to build on our alignment exercises as well as our haplotype painting exercises, I've just come up with a random sequence here for our mitochondrial Eve. Now, I want y'all to come up with 3 different haplotypes, an L3, an M, and an N. Assuming that L3 derives from our mitochondrial Eve, and again, pause the video here, try it on your own, reading through the directions, but I'll just walk you through in a sec. Since L3 we know derives from mitochondrial Eve, and we can distinguish them, we can go ahead and assume that the vast majority of the sequence is the same. L3 might be an A with an A, but then let's mutate a C there. No consequence other than to be able to distinguish them. Now, M and N are derived from L3, but they have distinct mutations that separate them. So they're going to be built on this haplotype. So I'm going to go ahead and copy our L3 type into our M and our N. But then I'm going to want to mutate different locations in each of them and switch bases. So maybe our M type would get a T to begin with, and our N type, switching colors here, might instead end with a C. And now when we're performing an alignment between these three, oop, didn't copy that piece. Ooh. We can look at this alignment, compare it to our original sequence, see that most of the bases are shared, that there's lots of agreement in lots of sites, but that they have distinct differences that separate them. And so in this way, by making up your own example problem of haplotype painting and inferred ancestry, you hopefully are summing up a lot of the computational biology that we've discussed in our last few labs and lectures. So the last task for today is to explore your own human mitochondrial haplotype. Where do your parents come from, particularly your mother? What haplotype groups come from that region? So, have some fun with it. Do a little bit of reading and make sure that you have a good grasp of our haplotype painting from today, and then also how we can weave in our understanding of mitochondrial diversity to tell us about human migration in the deep past. Well before humans were able to write, our DNA was recording our history for us.